Hello everyone, welcome to our YouTube channel Microbe Academy in the series of types of malarial parasites. In the previous video, I have explained in detail about the Plasmodium, a malarial parasite, how the parasite completes its life cycle, pathogenesis, clinical feature, diagnosis, prevention, and control of the parasites. If you have not watched the video yet, please go to the YouTube channel and watch the video, and I will also drop the link in below. And today we are going to discuss about the Plasmodium vivax. As I already mentioned in the previous video, there are several more than 150 species of the Plasmodium, and among them, four of them are responsible for the cause of the malarial disease in a human host, and among them one of them is a Plasmodium vivax, which has a widest geographical distribution extending through the tropics, subtropics, and temperate region, and it is believed to account for 80% of the all malaria infection, and it is the most common species of malaria parasite in Asia and America, and responsible for the cause of the benign tertian malaria. This is the overview and general global distribution of the parasite. As I already mentioned, it extends across the tropics, subtropic, and temperate zones, and is most prevalent across the South and Southeast Asia and Americas, but much less common in sub-Saharan Africa. And fever, as it causes the benign tertian malaria, where the fever spikes every 48 hours in a tertian pattern, and it is called the benign because mm it is called as a benign but can still cause a significant illness and of frequent relapses. The sporozoids of Plasmodium vivax are narrow and slightly curved. On entering the liver cells, the sporozoids initiates the two type of infection. Some develop promptly into the erythrocytic C zones, while the other persist in the dormant stage for varying periods as a hypnozoids, and there may be two distinct type of the sporozoids. The trachysporozoids, where the trachy means fast, they develops promptly into the primary exoerythrocytic C zones, and the next one is bradysporozoids, where the brady means the slow, which becomes the hypnozoids. These hypnozoids are the sleeping forms responsible for the relapse of the malarial fever after a month or years later. The pre-erythrocytic schizogony of the Plasmodium vivax lasts for the 8 days, and the average number of the merozoites produced per tissue season is 10,000. And the merozoites of the Plasmodium vivax preferentially infect the reticulocytes and young erythrocytes, and all stages of the erythrocytic schizogony can be seen in the peripheral smear. The degree of the parasitization is not generally heavy, and each infected red cell usually having only one trophozoite and not more than 2 to 5% of the red cell being affected. And the next is the trophozoite stage of the parasites. And the trophozoite stage of Plasmodium vivax is motile, and it form a ring form is well defined with a prominent central vacuoles, and the one side of the ring is thicker and the other side is thin, and nucleus is situated on the thin side of the ring, giving a particular signet ring appearance, and the ring is about 2 to 3 micrometer in diameter, appearing giving about a third of the size of an erythrocytes, and the cytoplasm is blue and the nucleus red in the stain films, and the ring develops rapidly to the amoeboid forms and accumulates the malarial pigment. The infected RBC is enlarged, and the infected red cell is noticeably larger than the normal cells, and the Schiffer's dot or red granules on the cell surface is the key diagnostic features of the parasites. And they become the irregular in shape, lose their red color, and present as a washed out appearance. and the few of them are parasitized, usually one trophozoite per cell, and maximum or 2 to 5% of the RBC are affected, and the malarial pigment in the ring stage turned and the ring stage of the parasite turn amoeboid in shape and accumulates dark pigment granules known as a malarial pigment. Next is the season stage and erythrocytic season. The season appears in about 36 to 40 hours and it occupies the whole of the enlarged RBC. The season matured in the next 6 to 8 hours with the development of merozoites and each with its central nucleus and surrounding the cytoplasm. The pigment granules agglomerate into the few dark brown collections at the center and with the merozoites around it, and this stage is present as a particular roseate appearance, and there are about 12 to 24 of the merozoites per season. And the erythrocytic seizogony takes approximately 48 hours, and the red blood cells which now measures about 10 micrometers in a diameter is heavily steeped and often distorted, and it bursts to liberate the merozoites and the pigments, and this pigment is phagocytized by the reticuloendothelial cells. And this all stage of the erythrocytic seizogony can be seen on a peripheral blood smear, which is the feature of the malaria, diagnostic feature of the Plasmodium vivax. This is the overall summary of the Plasmodium vivax. Several stages. We can see each stage in the primary blood smear of the Plasmodium vivax. Here we can see the erythrocytes and the young ring stage, and the infected erythrocytes are enlarged with a particular ring stage, and a mature adult ring stage enlarged in a RBC with a Scrivener's dot which is marked, and the mature seasons of the Plasmodium vivax shows a particular roseate appearance, and the male and female gametocytes of the Plasmodium vivax are also shown in the figure. Usually, the quickly summarizing, the RBC is enlarged with a Scrivener dots, and the gametocytes appears within a 4 days of the trophozoites. It causes the hypnozoites present in the liver causes the relapse of the malarial fever, and the parasite is responsible for the cause of the benign turtle malaria. The Plasmodium vivax cause the benign turtle malaria with a fever every 48 hours, but it still causes the serious disease and frequent relapses. The hypnozoites are dormant liver stage parasite that survive for a month to years and are responsible for the relapse of the malarial fever, and the infected RBC are enlarged. They show the Scrivener's dot and have a washed out appearance, and these are diagnostic clue for the diagnosis of the Plasmodium vivax. And the season stage produce 12 to 24 merozoites in a rosette pattern after the 48 hours, and all the erythrocytic stages are visible on the blood smear as I shown in the figure, and the gametocytes appear within 4 days and are the sexual forms that continue the life cycle inside the Anopheles mosquito. Thank you for watching. If you found this video useful, please don't forget to like, share, subscribe and press the bell icon. If you have any queries, please let me know on the comment sections. Till then, see you in the next video on the series of types of malarial parasite. Thank you.